Ah, it's that time. Ekis, 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 ekis. From the Llano Estacado, the Rio Grande Valley, from the bayou to the bottomlands, and from the desert to the rolling plains. This is a seminar for a wide range. Today on Soil and Crop Sciences Hour of Power, we have a speaker hailing from the great sandbag producing region of Weslaco. He's a native of the great meat on a sword producing region of Brazil. Dr. Jessup, would you like to introduce our speaker?
Russ, you're muted.
Can you hear me, Scott?
I can hear you now.
All right, I apologize to the group. Dr. Dasilva needs no introduction, but he's been with us for many years. He's focusing on polyploid breeding and genomics of sugarcane. That's expanded into other crops, wide hybridizations with sorghum species, miscanthus, and a lot of that has extended into the world of in vitro propagation, micro propagation that's useful across diverse crops. And I believe he's going to present today some of the utility of the bioreactors in potatoes. Dr. Dasilva.
Thank you, Russ, and thank you all for attending this workshop. On my presentation today, let's see if I can share my screen. Can you guys see my screen?
I can see your screen just fine.
All right.
Yeah.
Before I start, I just like to say that, as Russ said, on my nearly 19 years at Texas-A&M here in Weslaco, I have made a lot of presentations on different topics, but all of them were in person. So this is my first time doing this online. So please bear with me if something unexpected happens, if I press the wrong button or something. I'm trying to put in the movie here on the slideshow, but it's not going.
Harry, can you click on that little icon up there?
Yeah.
1, 2, 3, 4, 5, 6, 7 from the right or left?
Yeah, that's what I'm doing. I'm clicking it, but nothing is happening. Let me close the PowerPoint and
How
launch
about
it.
the one on the top? You're on the one on the bottom, aren't you?
Oh, yeah.
There's the same icon there.
Right here?
Yeah. I don't know.
No. Yeah, let me close this thing.
If you go to the very bottom, there's a PowerPoint thing next to the scroll bar, the Zoom bar, and you can press that and it'll go into presentation mode.
It didn't though.
It didn't?
You tried that.
No, I missed that.
Can you guys see this?
We can. You can also expand it and make it just slide it over.
I think I know why. I have another monitor here. Let me turn the monitor off. I think because it's going to the other monitor.
I think it should be okay. Later tomorrow, but it should be okay because everyone in front of the computer.
Yeah, let me launch it again.
Okay. Computers are so naughty.
Yeah. Are you guys see?
Beautiful.
Perfect.
Okay.
Oh, very good. Excellent.
Thank you. I told you that was my first time doing this. I knew something wrong was going to happen. Anyways, as Russ said, my presentation today will be on application of bioreactors to plant breeding. I work in collaboration with Nora Gracia, he also from the Weslaco Center, Isabel Valles, the A&M potato breeder, her graduate student Sanjeev Gautam, and Kranti Mandari, also here from the Weslaco Center. And what I'm going to do is I'm going to start showing advantage and disadvantages of bioreactors, what they do and what they are used for. Then I'm going to mention the mass propagation of energy cane that we did a few years back on the Chevron technology venture, as Russell said. But the focus of my presentation today will be as a new application of bioreactors for the screening of stress resistance in potato. Then I'm gonna mention a few future applications, screening for other stresses in potato, and maybe using RNA-seq to study gene expression, as we did with energy cane. Then I'm gonna mention some application also on Texas native plants, industrial hemp. I think Larry is gonna like to see that, and Russell also. And another possible application with switchgrass. Well, temporary immersion systems, or TIS bioreactors, as people call, is based on the twin bottles principle of two connected vessels. On the top one, there's a vessel that holds 6 liters of culture volume. It's called the plant vessel. And the bottom one, can you guys see my mouse pointer here?
Can see it, yes.
Yeah, so in the bottom vessel is what they call the media vessel. And that holds 3 liters of media. So those bottles are connected with a tube, and a number of vessels can be connected all together to air compressor, and which is set up to start from time to time. The air compressor pumps air into the bottom vessel. That pushes the liquid media up to the top vessel and stays there for a few minutes, like usually 15 to 20 minutes. Then the air compressor turns off and all the media drains back by gravity, and which is very efficient because that drains all the liquid from the top vessel. And it works pretty well. So basically, the advantages of this system is that it represents a high productivity and efficiency process. It also allows a better control of cultural conditions, supply optimum nutrient hormones for plants, and is an automation process, allowing a better cost benefit as compared to the other mediums, the other propagation methods, micropropagation. There are a couple of disadvantages. One is microbial contamination, but this one I left with a question mark because in our case here, it rarely happens. It's very difficult to find contamination. And also, but unfortunately, there are some species that are recalcitrant. You cannot micropropagate them with this system. But so far, all the species that we try, in addition to sugarcane, energy cane, we tried miscanthus, miscane, and other sugarcane-related species. Now, recently, we've been working with Amelia patens, another species, potato, as you're gonna see, work beautifully, and so on. So far, I haven't had any problem. Even hemp, we start playing with it, and so far, I haven't found any species that is recalcitrant. Anyways, just to give you a picture, this is all energy cane. And as you can see, as I mentioned, they're all connected through tubes. Those are filters to avoid contamination. If one of those got contaminated, it doesn't spread to the other vessels. And all of them are connected to here to this air compressor that has a timer, and we can set up to start. Usually here we do 4 times a day, 24/7. It pumps air there, it stays for 15 minutes, then turns off. All the liquid media drains to the bottom vessel. It stays there. 4 hours later, it goes up again and stay there 24/7. Here's another view of the bioreactors, but now those are LEDs. It looks pink because in this case there's only blue and red wavelengths. That's all plants need. So, and by doing so, it takes less energy and doesn't heat as much as light. So it's cheaper and more efficient. So this is a typical bioreactor from energy cane. In 3 weeks after we inoculate one single seedling in this bottle, you can come up with up to 800 plants. So that's pretty high. Just to give you an idea, it takes, at this rate, it takes only 7 bioreactors to fill up 1 acre of energy cane. That's approximately 5,800 plants in 3 weeks. And just to give an idea how big this is, the natural rate of multiplication just by planting seed cane with seed pieces, it is 1 to 6 here in the US. In Brazil, under optimal conditions for cane and the tropical is 1 to 10, but here is 1 to 6. So, and this rate with bioreactors, this rate is 1 to 1,200. So the area that you can produce using this system is 200 times bigger as compared to the natural condition. And when we used this for Chevron, they were really happy. It was highly efficient to do big success. And after those 3 weeks, we take those 800 plantlets or seedlings. You have 2 options. You either inoculate another 800 vessels, or you can take those to the greenhouse. And here is just a picture of those seedlings after 1.5 months to 2 months in the greenhouse, the energy cane. Those plants are ready to go to the field. And another advantage as you can see here on the left-hand side, those, if you plant those in spindling trays, they can be planted mechanically. And afterward, irrigation. Irrigation is a plus. It's too risky to wait for rain, because those seedlings are really sensitive to lack of humidity. But after it's established, it's a normal gain. And just a picture, I believe those are 4 months old seedlings, energy cane seedlings. Like I said, we produce a total of 120,000 seedlings of energy cane for Chevron, using just this. And we have recently published a paper, GG actually, on bioreactors in vitro cell development biology plants. That's one of the best journals for micropropagation. There's a DOI number there, the reference if anybody wants to. There's open access. Now, enough of sugarcane, let's switch to potato. Those 2 plants are very different. Cane is an interspecific hybrid, highly polyploid, 12 copies of chromosomes each. And whereas potato is a tetraploid, only 4 copies. And cane has a huge genome. But even though they have all these similarities, they are both highly heterozygous and can be propagated vegetatively. And also by botanical seed. So in such species, the first step of breeding is make hybridization crosses. And they are cross-pollinated, so all the varieties are heterozygous. The first stage of the breeding selection is heterozygous population of seedlings, resulting from the cross-pollination among the genotypes, the parents, used as parents. And then there's visual selection, which includes a lot of subjectivities, as you guys know. But from there on, all the plants selected are clonally propagated, so we fix the genotype. And depending on the crop, there's a number of successive stages, always with visual selection and clonal propagation, until the number of genotypes is reduced enough so one can plant a competition trial with replicates for statistical analysis and yield assessment, which reduces the subjectivity. So when we all started from sugarcane to potato, Isabel Valles went to visit us here in Weslaco a couple of years back. That was before the COVID deal. And when I showed her the facilities that we had to use in sugarcane breeding, she immediately saw the potential for doing this. Use bioreactors to screen for stress resistant in potato. This is just a blow up here to show you bioreactors with those actually potato plants inside the grow chamber. So you can control the temperature you want to keep those plants under here. And then we set up a set of bioreactors under optimal conditions, 16 degrees, and a set of bioreactors with the same genotypes inside grow chambers under heat stress, 30 degrees. So speaking on heat stress in potato, the heat can produce external effects, as you can see here on the left, and also what they call knobs here, also external effect due to heat. On the right hand side, there's Atlantic, one susceptible varieties that shows internal heat necrosis. Okay. Now, Solanum tuberosum is one of the major crops of the world, being the most efficient food crop in terms of producing human calories for human consumption per unit area. But high temperature leads to biomass allocation shifting. In addition to the damage, it also shifts the biomass production from tuber to leaves. And if that happens at the early stages, that could lead to a drastic yield reduction, and in addition to quality deterioration. Whereas if that happens later in the tuberization, high temperatures could lead to quality loss and production of less marketable product, which is also big damage to the grower. But luckily, there is genetic variability among the potato germplasm. Some varieties, they do behave differently to heat stress. And that's what we want to explore. So in this context, Texas has recorded among the highest summer crop yields in the whole US, among the other 9 states that produce summer crops. So if we can come up with tolerant genotypes of potato, that will be a big deal for the potato industry in our state. So, but when it comes to screening for heat stress, we need to set up the optimum screening temperature. And on this panel here on the top, you can see Atlantic, susceptible variety. When they set up, there was Isabel in her group, set up the 35 degrees centigrade, nothing grew. But at 16 degrees, which is optimum, everything works fine for both Atlantic and Russell Burbank, but nothing at 35 degrees. So we need to know the exactly temperature for screening. Now, screening under field conditions is difficult because there's a number of factors that mimics and makes trouble, confounding effects. So in vitro screening in potato has mainly two advantages. It allows a screening of a large number of genotypes, and is also free from confounding effects from the field. So our strategy was to produce media, in vitro media for microtubulization in solid media in College Station in Isabel's lab, and liquid media here in my lab using the bioreactors. And then we set up the heat stress temperature at 30 degrees to screen for the microtubulization and test the genotypes to see which are acceptable, which ones are tolerant. So, but percent microtubulization is variable with solid media. In liquid media, the microtubulization is difficult because it requires to change the media very often, which leads to increase the chance of contamination. But that's the advantage of using bioreactors. That's what Isabel realized. Because with bioreactors, you reduce the need for changing, or at least you can change the liquid easier with less risk of contamination. Now, switching to field evaluation, that's Isabel's group. They did last season, 2019-2020. Two varieties there. A acceptable variety, Russell Burbank, which is prone to external deformity, and Atlantic, which is prone to internal defective. They both behave as expected, and one of the Texas bred clones, the Vanguard Rosette, that we call TX08532, was consistent in producing marketable tuber with less external as well as internal defects, which is good news. And as you can see here, there's a big variation on the marketable yield, and also the percent of tubes affected by defects among the genotypes. So we set up the bioreactors in Weslaco. That's a shot from one of our grow chambers with the bioreactors all connected with the tube coming from the air compressor, as I showed you. And we set up those grow chambers at 30 degrees centigrade just to screen the microtubulization of each genotype. So just one comparison here, Atlantic on the top, this is a susceptible variety, and this clone from Isabel and DTX, and the bottom was expected to be tolerant, and sure enough, when you look at the 16 degrees centigrade, the optimum temperature, both produce beautifully in bioreactors, and at the heat stress, the Atlantic produce much less. Both produce less, but the Atlantic showed a reduction much more intense as compared to the NDTX clone, which was expected to be tolerant, confirming what we expected. So another shot, also still comparing the same two varieties, and the susceptible one showing more damage under hot conditions as compared to the other one. But it was interesting to note that this NDTX tolerant clone produced some microtubes at the stem, which was surprising. It may be the way how this genotype express his tolerance to heat. The same two genotypes again, on the optimal conditions, as you can see, the microtubulization and how stress reduced in both cases, but reduced much more in the acceptable one as compared to the tolerant one at the 30 degrees centigrade. Now, the bottom line is when it comes to compare these two varieties, if we express the reaction to hot temperatures in microtubules per dry weight, you can see that the susceptible one, Atlantic, produce only 1.5% microtubes as compared to what it produced under optimal condition. That's a big reduction. The other one, NTDX, also reduced his microtube production, but produced 8.6% of the microtubes, which much less reduction than the susceptible one. It's 6 times more intense, the reduction here on the susceptible. So this proves that for these two genotypes, one can use the liquid media stress, heat temperature with bioreactors to identify which one is cell tolerant, which one is susceptible. Now, again, the same two genotypes, Rosette. Oh, no, I'm sorry, those are two other genotypes. Now, Rosette and ATX, another clone from Isabel. The Rosette is the susceptible one. See, at the optimum temperature, 16 degrees, produce lots of microtubes, and so does ATX. When you switch those to 30 degrees under heat, the susceptible one reduces the microtubes. The tolerant reduces also, but the reduction is less. It's not as less as the previous one, but it's still less. Another shot here, the same two Rosette at the optimal temperature, ATX at optimal temperature, producing lots of microtubes and less in the under stress. One important thing also here on this ATX tolerant clone is that it produces a larger microtube, as you can see here. That was interesting. It was unexpected, and also it may have to do with the way this genotype handles the heat stress. But again, once we express the microtube or the tolerance to heat on microtubes per dry weight, the susceptible present a reduction much more intense than the tolerant one, as you can see here. And also, you can see the bigger microtubes under stress as compared to the other variety, and also to the same variety under optimal conditions. So it looks like for this particular genotype, the stress leads to bigger microtubes. And also here, expressing them in terms of microtube per dry weight, the reduction was not as extreme as the previous two other genotypes, but as you can see also, the reduction was 2.4 higher or more intense in the susceptible variety as compared to the tolerant variety. So again, the bioreactors proving to be efficient in terms of screening heat stress in the case of those two genotypes. Now, one interesting also is here, this ATX clone from Isabel showed much bigger microtubes, almost mini tubers, as you can see here, as compared to the same varieties under the optimal condition, 16 degrees. It's something that is intriguing. So now that we are happy with using this as a screening methodology for potato, we intend to publish this data and try to use this data as preliminary results for the grant proposal. And in addition to potato, or at least to heat stress potato, we could also use this methodology to screen for other stresses, such as drought and salinity, which, according with the literature, also have severe adverse effects in potato. Because the composition of the liquid medium, we can mimic those stress, both drought and salinity. We can actually screen different genotypes for tolerance or reaction to those stresses. And that would be advantageous for a breeding program. And we also could use this methodology to do some differential gene expression analysis through RNA-seq, for instance, as we did with energy cane and sugarcane. So in that case, what we did was, there was this wild sugarcane called Saccharum spontaneum, which is considered also a noxious weed. I have it here in Weslaco in my farm, but in order to plant that variety, I have to get a permit from the USDA, and they only give this to if you had to take a lot of care to get the permit. So what we did was we introduced this Saccharum spontaneum, which has tolerance to cold, and we did some RNA-seq with that species, the wild species, and the regular sugarcane. And we detected around 200 genes that were upregulated with the cold stress. And we were able to identify 7 individual genes after 20 hours at 5 degrees centigrade. 5 degrees is pretty cold for sugarcane, a tropical plant. Not as cold for potato, but it is for sugarcane. And the QRT PCR confirmed 4 genes that were involved in cold resistance. And one of those genes, our aquaporin gene, we use as a proof of concept. We transformed tobacco, a model plant, and the same gene confer resistance to drought and salinity, in addition to the cold. As you can see here, that's the the tobacco plant, wild type here, with showing symptoms of drought, and the plant transformed with the wild cane, aquaporin gene, showing some drought tolerance. Same thing with salinity. That's the wild type tobacco plant here, and those who are the same genotype, but with the same aquaporin gene covering some salinity. So if the same thing happens to potato, once we identify genotypes resistance to one of these stress, there's a good chance that those genotypes will be resistant to the other stress as well. But now switching from potato, one feature application of this methodology is with Texas native species. Little Bluestem is one of these Texas native grasses. We just submitted a grant proposal with Jim Muir from Stephenville, Texas AgriLife. And in this case, there's Little Bluestem for those of you who never saw one. It's one of our next Texas native ornamental plant. And when we tried, that's the first time we tried this species in bioreactors. And as you can see, not only the plant grows well, but also there's the regular media, liquid media, and this here is within bioreactors. As you can see, after the same time, in bioreactors, the production is much higher. So Little Bluestem can be actually more efficiently produced in bioreactors also. Another potential application could be used for industrial hemp. The hemp growers say that homogeneity of variety in each variety is a problem. Been in touch with some colleagues from LSU, and they say that sometimes when two people are talking about the same variety, they are talking about different genotypes because it's very heterogeneous. So clonal propagation would solve this problem. In addition, some growers also like to propagate hemp with what they call stakes, just clonal propagation. And apparently in the literature, the clonal propagation for hemp CBD is already well established, but there's a lot of work to do for hemp to produce fiber. And so there's some room there for research. And those pictures are from my lab. We just started doing some micropropagation of hemp in bioreactor. Now, another potential application could be use this methodology with switchgrass. According with the switchgrass breeders, the plant doesn't have any lateral buds like sugarcane. So all the propagation has to be through botanical seed. And if we were able to propagate switchgrass by tissue culture, by cloning, that would, according to them, the breeders, could shorten the cycle by at least 2 years, which would be very attractive for any breeding program. So I think with this, I've reached the end of my presentation. I'd like just to acknowledge the support of the Texas A&M AgriLife Research and Extension Center in Weslaco for providing the funding for this research. And with this, I open to the floor for any questions anybody may have.
Great job, Jorge. Great job. Questions?
Thank you.
Oh, is that you, Peyton?
Yeah, just responding to your chat.
No questions. I see. That's like an applause sign. Great.
Oh, yeah.
Very good.
Thank you.
Questions? Fire away.
I hope I didn't went too fast. I was told to use 50 minutes. I don't think I used that many.
You're a speed demon. So there's a question in the chat from Shakira. I can read it to you if you like.
Okay, please. Appreciate it.
How do you manage contamination within the bioreactor?
That's a good question. But one, like I mentioned, I'm sorry. Well, like I mentioned, as I showed, we have filters to isolate in each one. So if one happens to be contaminated, it doesn't go to the others. They are connected. But we mainly, I guess, I attribute the low rate of contamination in my lab to my technician, Nora Gracia. She went to Brazil for a while to get a special training there, and she really came back with a hang on the issue. And so one thing that I know she does is we start from material that is very clean. We usually keep plants in the greenhouse as much as clean as we can. And before starting the tissue culture, she does a set of cleaning with Clorox in different concentrations in alcohol and all that. So yeah, so far we've been able to keep contamination very low. Like I said, even the BP lead scientist was surprised to see our low rate of contamination.
Other questions?
Yeah.
Well, I have
a question.
Oh, go ahead, Sakiko.
Sorry. So I might have missed it, but what is your starting material? Are you starting from the side shoot or are you starting from tissue culture?
No, actually, the starting material is actually, in the case of energy cane, was from the lateral buds.
Okay.
And also, you can take the meristem tip of the plant. If you want the plant free of disease, it has to be from the meristem tip, right?
Okay.
But if you don't need to be, if you're happy with your material, if you're not specifically using tissue culture just to clean from disease, you can use from the lateral buds.
Okay.
In case of sugarcane, in case of switchgrass, for instance, there's no such lateral bud, so it have to be the meristem tip. But you can be, the tissue culture, the starting material can be from a real plant or in the pot or even in the field. If you're able to clean up the material well to reduce the contamination, that's why the starting is from the plant itself.
Okay, thank you.
This is Isabel. Let me add. Sorry.
Go ahead, Isabel.
Yes, I would like to add about potatoes. In potatoes, once the material looks very promising, we bring the tubers to the lab, and then we conduct ELISA tests to figure out if we have presence of viruses, and if so, we apply chemotherapy or thermotherapy to get rid of the viruses. And that is what we keep in tissue culture. So what we keep in tissue culture is disease-free material. And those are the type of materials that we provided to Jorge and his group. So they received plantlets in tissue culture that were disease-free, and then they propagated those 2 years in the bioreactors.
Thank you, Isabel. I didn't know that, but.
Other questions?
Do you work with strawberries?
One at a time, people, one at a time. Go ahead, Dr. Baltensperger.
Do you work with strawberries?
No, we have never worked with strawberries.
Okay.
But we're always open to new crops. If you know any opportunity, let me know.
Zhenlan, were you trying to ask a question?
Yeah, I want to ask a question about the qRT-PCR. I found you use that way to find some gene. I want to ask, how did you find those genes? Because if you use that way, there are many genes expressed during your experiment.
Well, what we did was, as I said, we did the RNA-seq. We went to Charlie Johnson at Texas A&M. He ran the RNA-seq for us, and we were able to see those genes differently expressed. And at that time, I had a postdoc. He was the one who did all the bioinformatics, and he came up with those 7 genes that he ran the RT-PCR. And once he confirmed the RT-PCR expression of those 7 genes, we isolated 4 of those. And the one that I mentioned, the aquaporin, was the one that was more prominent. The differential expression analysis, once the cane was exposed to cold, the expression went very high. And that's why we target that one, and that's the one that we use to isolate and transform tobacco as the proof of concept. But the other 3 genes that we isolated, because at that point we end up the support finished, the granting. The guy went back. He was from Brazil. He went back to Brazil. So we published a paper, but the research finished right there. But most likely, if the other 3 genes that we extracted could also be has a effective in those stress as well. like not only in cold, but also salinity and drought. I wouldn't be surprised. Because that's what the plant physiologists say. What the mechanism that protects the cell membrane from cold, usually the same mechanism that protects the cell membrane from drought and salinity, for instance. So if we find a gene that confers this protection to one stress, it usually provides protection to the other stress as well.
Oh, okay.
I would be surprised.
Thank you so much.
Hello? Scott? You hear me? Hongbin.
Yeah, go ahead.
Yeah, I have two questions, Jorge. One first question, did you did any experiment to make comparison your data from the reactor, bioreactors and the field data?
I'm sorry, can you say that again?
What's the, you have any correlation the data you collect from bioreactor with the field data? Let's say for heat tolerance or drought tolerance, whatever.
Well, the field experiment was done in College Station by Isabel's group. Here, we only did the material, the propagation, and the screening in the lab with the grow chambers. So the field screening was made by Isabel. Maybe she can answer.
Okay, so actually, the locations where we have field trials are in the Spring Lake and Dalhart in the high plains. Potatoes don't grow in College Station. We only have them in greenhouses. Over many years, we noticed that since in Texas, in the high plains, we definitely have high temperature stress. And by looking at marketable yield and internal and external defects, we've been able to identify clones that perform much better. So a small set of those have been tested over a period of multiple years, and we identified contrasting clones, some that perform pretty well and some that have lots of issues with reductions in yield and higher percentages of internal and external defects. So one of the first ideas was to identify and confirm checks for follow-up experiments. So identifying kind of extremes. And actually that's why I ended up sending to Jorge Russet Burbank, which is the most widely processing potato grown in the US that is very susceptible, has lots of deformities, external deformities. So that would be one of the susceptible checks for the russets. And then for the chippers, Atlantic is one of the most widely grown potato chippers and is susceptible to high temperature, but mainly because of the internal defects, the internal heat necrosis. And then the other two clones that he tested are actually Texas selections. One of them is Reveille Russet. He still used the experimental name, but it's Reveille Russet. And the other one is, so the Reveille Russet would be a russet type. And then the other one that we declare as potentially tolerant. And the other one is a Texas selection that is a chipper that we declare as potentially tolerant chipper. So we wanted to have like contrasting sets for the russets, contrasting sets for the chippers, with the idea of later on do some type of RNA-seq experiments to understand why the tolerants are actually tolerant. And we could definitely use also a screening to follow up, unselected material and do other studies. But the initial thing was, let's be strong about what we declare susceptible and resistant, and then let's understand why, there is that contrast.
Thank you.
Oh, there's one from Steffi in the chat. In the case of potato, approximately how long it takes to start seeing microtubers formation, and how long are the plants left in the bioreactor before collecting them?
Well, we start seeing microtubers here in bioreactors after 1.5 to 2 months. That's when we start seeing them. But we left longer just to make sure they will produce enough microtubes. So what was the second part of the question again, Scott?
How long do you leave them before you collect them?
Oh yeah, well, we just started this year, so we don't know yet what's the optimum time for leaving them. We harvested those because we want to publish a paper, like I said, and our goal is to collect all the data by the end of this year and see if we can submit something early next year. So actually, we don't know yet what would be the optimum time to maximize the microtubes. What we want to do now is just to have evidence that the bioreactors can actually be used and confirm the resistance to the stress that there is no, like Isabel mentioned.
So as it is, it doesn't look like they'd provide a very satisfying meal. Maybe you'd have to grow them a little bit longer to get something that's, more human-sized rather than mouse-sized.
Yes, well, the microtube, I guess Isabel can explain better than me, but the microtube is just like a artifact to see that it's highly related to actual tuber production. So the production of microtube should be strictly related to produce our actual tubes in real life.
So you don't eat them?
I guess not. I guess just if you plant microtube, will they germinate, Isabel?
Actually, in the context of the seed certification system and seed increase in potatoes, everything starts in tissue culture. So from tissue culture, there is propagation, and then in the traditional system, the materials are moved to the greenhouse and they are grown in small pots. The size of the tubers is typically related to the size of the container where they are grown, but it is desired that in the greenhouse, we produce what is called actually mini tubers. They are a little larger than those that were shown, mini tubers, but the mini tubers that are disease-free are the first generation of the certification process. You plant those mini tubers in the field and you get regular sized tubers. But it's much easier to handle as the first step, really small tubers as a way of propagation. I also would like to mention that the beauty of the bioreactors is that since the media is liquid, it allows to switch between the optimum growth media in which the plants will be grown for like a month or so, and then switch to tuberization media, which would have a much higher level of sucrose. So that's something that is special about the bioreactors. And then that also in the context of the contrasting temperature, so you can see what happens under optimum conditions versus high temperature stress.
Yeah, good point.
Yeah, so the microtubers, if you plant them, you would end up with plants. And then those plants, if they are grown in, large pots or under field conditions, they will produce the type of tubers that you are used to see.
Yeah, that's a good point. As Isabel mentioned, in the case of sugarcane energy cane, we also can play around with the composition of media. When I mentioned that once you have those 800 plantlets in one bioreactor, at that point when you harvest them, you can have the choice to put another bioreactors or take them to the greenhouse. If you're ready to transplant at the greenhouse, we switch the media to rooting media, like she said. And then the next step will be to harvest to the greenhouse. If you want just to produce, to inoculate more bioreactors, and then we keep on the same media, and you won't produce much root. So the bioreactor allows you to just flexibility, or the kind of infrastructure or the plant structure that you want to have.
The other aspect is light. The optimum photoperiod for vegetative growth is 16 hours of light, 8 of darkness. But the moment you want to induce tuberization, you switch to 12 hours of light, 12 hours of darkness. So the switch to go from vegetative growth to, like the tuber production, it's by changing the type of media, but also modifying the photo period. And those two things, are feasible in the context of the bioreactors, together with controlling the LED lights and as well as the temperature. The temperature is another important aspect, because in potatoes, when the let's say the night temperature, it's under 20 degrees C, there is promotion of tuber formation. So there is a storage of starch in the tubers. But if the temperature is above 20 C, then there is not much tuber formation. So by looking at all of these parameters, light, temperature, and media, we are able to switch from, let's say, just above ground growth versus storage of carbohydrates in the tubers.
Very good. Other questions? All right. Well, thanks again, Jorge. You did a good job.
My pleasure.
Very interesting to hear about the use of bottles and compressed air and plant propagation and potato breeding.
Indeed.
Thank you very much.
Thank you to you, Scott.
Thank you.
Thank you. Good
Thanks,
job.
bye.
Bye-bye.
See everybody, same channel, same time next week.
All right.